This video covers drawings for the urinary system, and we'll start with an overview of the kidney. Here is a section of the kidney. This is the capsule. And the kidney has two regions, a cortex and a medulla. In the medulla, there are structures called renal pyramids. And I'll draw a few of the renal pyramids here. The tips of the renal pyramids end in what's called a renal papilla. And the filtrate drips out of there into a little cup-like structure called a minor calyx. Draw another minor calyx here. Where two or more minor calyces come together makes a major calyx. And where even more come together in the middle of the kidney is the renal pelvis, which sits in a space called the renal sinus. There's also a little bit of fat in the renal sinus. Between the pyramids are the renal columns. Now, where the business of the kidney really happens is in structures known as nephrons. And there are two different types of nephrons that function slightly differently and also have slightly different locations. The cortical nephrons have their glomerulus quite far into the cortex, and their loop of Henle goes down into a renal pyramid. So this is a cortical nephron. This loop of tubing is a loop of Henle. And this is the, actually really the renal corpuscle, which holds the glomerulus. The other type are juxtamedullary nephrons, and their loops of Henle go very deep into the renal pyramids. Juxtamedullary. That's a U nephron. The juxtamedullary nephrons are involved with making the urine more concentrated. That's what they specialize in. Our next drawing is the filtration membrane. This is inside the renal corpuscle, and this will be the layers that the filtrate actually passes through. We'll start by, in a lazy way, drawing some simple squamous looking cells, although these are actually the capillary endothelium, and these are fenestrated capillaries. So you may recall that we used those little channel-looking things to indicate fenestrated capillaries. And we could draw a red blood cell in here just to remind us what's going on that side. Then the podocytes of the renal corpuscle have little feet and arms that are called podocytes and pedicels. So the podocytes are the cells, and these are the pedicels, the little foot-like projections that make a network over those capillaries. So there'll be many of these. And there would be a little bit of connective tissue, a thin basement membrane here. These are the pedicels of the podocytes. Podocyte are the cell type. The filtration slit is this gap between the pedicels, and frequently the fluid will pass through the fenestrations of the capillary endothelium and then between the pedicels and get collected in the Bowman's capsule. The next drawing is of reabsorption in the proximal convoluted tubule. And we're going to draw a few cells here. And there's cells on the other side as well, which I'm drawing less completely. These are cuboidal cells, and they are connected by tight junctions. And so what's in between is the lumen of that proximal convoluted tubule. Off to the right would be a capillary, and that would be a peritubular capillary. There are several substances we're interested in here. One of them is sodium. And sodium will enter these cells by diffusion along its concentration gradient, and that concentration gradient is maintained by sodium, potassium transport. And we've met that before. You know that potassium comes in, sodium goes out. Normally there would be 2 potassiums and 3 sodiums, and that uses ATP. Standard sodium-potassium pump, which helps to bring that sodium into the capillary. Another substance of interest is water. Water will tend to follow the sodium by osmosis. And that goes all the way across by osmosis. Chloride ions enter the cells by diffusion. And they pass on into the capillary by diffusion as well. And then glucose. Glucose enters those cells primarily by a sodium glucose symporter, in which 2 sodiums enter the cell along with a glucose. That glucose can pass out of that cell to be picked up by the peritubular capillary through a protein by facilitated diffusion. And our next drawing is about the concentration gradients and how they're maintained within the kidney. So here is a kidney, and here's the edge of the cortex to the medulla. And there is a consistent concentration gradient that is maintained within the kidney. The concentration is about 300 milliosmoles in the cortex. And as we go deeper into the medulla, the concentration increases to about 1200 milliosmoles. The osmol is a measure of osmolality as opposed to osmolarity. They're similar, but not exactly the same. And this concentration gradient is maintained because of the loops of Henle, especially the juxtamedullary loops of Henle. So now I'm going to draw a loop of Henle here. This is the descending limb. Loop of Henle. This is the ascending limb. So the fluid's flowing through in that direction. The descending limb is freely permeable to water. And it is not permeable to ions. The ascending limb is permeable to ions, but not to water. So things like sodium and potassium and chloride can exit in the ascending limb, but not the descending limb. Because water is being pulled out on the way in the descending limb, and ions are being pulled out in the ascending limb, that gives a higher concentration at the end. So we would have the 1200 milliosmoles here at the U-turn and about 600 milliosmoles at the top of that loop.